Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I'd like to thank all of you who joined us live at our first Dr. GPCR virtual cafe held on March 26th. Our next virtual cafe will be on April 29th at 11 a.m. EST. Our guest is Dr. Joanne Kamens, the Executive Director of AdGene. AdGene is dedicated to accelerating scientific discovery by enabling the easy, open sharing of high-quality controlled biomaterials, including those used in GPCR research. To reserve your free ticket, visit drgpcr.com/virtual-cafe today. To present your work or your company's technology, or to sponsor the Dr. GPCR Virtual Cafe edition, please fill out the form on the cafe's main page. The goal with the Virtual Cafe Talks is to bring you GPCR-focused live presentations every month. You'll be able to join us live and interact with the speakers and attendees. We hope you'll join us on April 29th. To stay on top of all your GPCR news, subscribe today to our monthly newsletter at drgpcr.com/newsletter. Last but not least, find us on YouTube and subscribe to our channel. On our channel, you can watch the latest Dr. GPCR podcast episodes and even the Virtual Cafe Talks. Come check out Dr. Brian Airy's presentation about GPCRs that are involved in modulating flow-induced signaling pathways in vascular endothelial cells. And now, let's dive into our episode. Hello, everyone. This is Yamina from Dr. GPCR. Welcome to another episode of the Dr. GPCR podcast. Today, I have the pleasure of having with me Dr. Kevin Pfleger from the University of Western Australia. Hi, Kevin.
Hi, lovely to be here. Thank you.
I'm so happy to have you here, especially we've been trying to schedule this discussion for a while, and we both have had I'd had some technical issues. I'm so happy to have you here. I was looking yesterday at the website of your group, and I saw that you have so many roles and responsibilities. How do you do it?
I have a great team. In fact, I have multiple great teams. So I think research and innovation is not for one person anymore. It's for teams of people, and I think it's about building great teams that you can depend on, and that's really the secret.
That's amazing. So for our listeners, I'm just going to list some of these roles. So Director of Biomedical Innovation, Head of Molecular Endocrinology and Pharmacology, Teaching Molecular Pharmacology of GPCRs and Molecular Neuroendocrinology, Chair of Accelerating Australia Executive Committee, and I could go on and on and on. But yeah, I think you nailed it. Science is a team effort. So how did you get to where you are today with all these arm-long roles?
So I started off in Cambridge, did natural sciences and majoring in pharmacology, went up to Edinburgh, the University of Edinburgh, did my PhD at the Medical Research Council Human Reproductive Sciences Unit, and then came out to Australia 18 years ago. Planned to come out for a couple of years, but never went back, which happens a lot when the English come to Australia. And had a number of nationally competitive fellowships. We've got a spin-out company that has been a great adventure, and that's just completed phase 2 clinical trials. And so really working on that boundary of academic research and translation, and very exciting, very exciting.
How did you get into science in the first place in the UK?
So I had really good teachers, fantastic chemistry and biology and physics teachers, and always had a passion for science, always wanting to know how the world works and why things happened. And so I went to Cambridge, and the great thing about a natural science course at Cambridge is that you can have quite a diverse range of courses and specialise up to the final year. And then the PhD was a great opportunity. In fact, I went across to Oxford to interview for a research assistant position, and the person I went to see actually sat me down and spent the whole time saying, "You don't want to be a research assistant; you want to go and do a PhD." So it was the most bizarre interview I've ever had, but it was great because at that point I didn't even consider doing a PhD, and it really started me on that path. So I think I've always been interested in science. I've always loved biology, and And since I went to Edinburgh, always loved GPCRs.
Tell us a little bit more about your first contact with the GPCR field.
So at Cambridge, we studied molecular pharmacology and systems pharmacology, and I was always more interested in the molecular aspects. And obviously GPCRs, such an important drug target, and important in so many different parts of physiology. So was very, very excited to be getting the opportunity to go to Edinburgh to do a PhD on gonadotrophin releasing hormone receptors, and yeah, never looked back.
That's amazing. That's amazing. A lot of us have this story where, you accidentally bump into GPCRs at one moment, and then you just never stop, and you can't let go. And then, was that your first, after your first encounter with working on GPCRs, did you decide that you wanted to continue in the field because you liked GPCRs so much, or was it kind of a natural evolution of your science?
I think there were so many aspects of molecular pharmacology. I mean, I came out of Cambridge where my lecturers taught me about biosignaling, and I went up to Edinburgh, and this was the thing about Cambridge. You were taught by the cutting edge researchers, and I went up to Edinburgh thinking that everybody knew about biosignaling. But this was back in 1998, and I don't think the term had even been coined yet. So we called it all sorts of different things, ligand-directed selectivity and all sorts of different terms. So that was an eye-opener to realize that I was learning things that people who were senior professors didn't know at that time in other parts of the country. So it was fantastic to know that I was working at the cutting edge, discovering things that nobody else knew. And that was the great thing about being a PhD student, was you're working on things that nobody else knew about, never seen before, and got to travel the world, went to conferences in the US and Europe, and just a great, and the people you work with as well, all on the same journey. And wherever you go in the world, you meet like-minded people, particularly in medical research, all striving to eventually improve a patient's life, and it's fantastic.
I could not agree more. I think having the ability to work with people who are like-minded, who have the same goals, and being able to go to conferences and meet these people. Well, we can't do it this year, but hopefully we'll be able to get together at conferences and talk GPCRs further out. So you're in Edinburgh, you decide to go to Australia. What led you to Australia?
Well, there was a professor in Edinburgh. In fact, I went to Edinburgh to do a PhD with Karen Edney, and she actually moved to Australia just before I got there. So I ended up doing my PhD under Robert Miller, and I always joked with Karen that well, you have to give me a postdoc position now that you've moved to Australia, and actually she did, and so I moved out to Australia again, only planning to be there for a short time, but loved it. Got a competitive fellowship, and then started working with Dimerix and never looked back. So it was the connection with Edinburgh that brought me across. But having established myself here, always keen to stay.
That's so interesting. I've always wanted to go to Australia. That's one of the continents I haven't been on. So hopefully one day. So then you're in Australia. You're doing a postdoc. What projects were you working on? Any specific GPCRs that you got interested in compared to your PhD training?
Well, the irony was that since coming to Australia, I've done a lot of work with arrestin interactions with GPCRs. And of course, the gonadotrophin releasing hormone receptor doesn't interact with arrestin, so it doesn't have a tail. So that was ironic. But I've always largely worked on endocrine receptors. Not entirely, but generally worked on endocrine receptors, being a molecular endocrinologist as well as pharmacologist. So I've been working on the angiotensin receptor, vasopressin, arrestins, moving more towards chemokines as well. So, but tending towards the peptide chemokine end of the spectrum.
Any preferred receptors or family of receptors in this list?
Yeah, as I say, mainly the angiotensin, but also the function interactions the angiotensin receptor has with a whole range of GPCRs. We've published not only with the type 1 and type 2 interacting, but also the interactions with a whole range of GPCRs, but also other receptors as well, tyrosine kinases and other receptors. So I think you had Thierry Hibbert on earlier, one of your podcasts, and
Yep.
I listened to that the other day, and absolutely I would echo everything he said. The angiotensin receptor seems to be pivotal in a lot of these function interactions.
Exactly. As you were speaking, I was thinking about the first discussion I had with Terry about the angiotensin receptors. I think it's a fascinating receptor. I've always worked on chemokine receptors or peptide binding, protein binding receptors. But the discussion with Terry about angiotensin receptor was really, really fascinating, and I think it's a great system to be studying.
Absolutely.
So, angiotensin receptor, that's one of your favorites. Can you tell us a little bit more? So the discussion with Terry was recorded sometime in February this year. Anything new happened in the field of the angiotensin receptor?
Well, I think there's little things happened since then, called COVID-19, and in fact, that with its interaction with the ACE2, it does actually have quite a role with the angiotensin receptor system, in fact, and we're only just understanding what that is. But in fact, we're actually looking to trial in the REMAP-CAP trial to look at the angiotensin chemokine CCR2. So with DMX-200. So yes, absolutely, the angiotensin receptor seems to be very important in COVID-19, and to the extent we're not quite sure yet, and there's a lot of work to be done, but it does seem to have a role. Absolutely.
I had a similar discussion with Paul Insel. We had a recording about COVID and what he was doing during these times. And the first discussion we had, I told him, I'm pretty sure there's a GPCR involved somewhere with COVID, and it turned out to be AT, the angiotensin receptor, in some form. I just remembered something. So getting back to your path, you had mentioned that you've been in academia, but also you've been working in a spin-off company. Can you tell us a little bit more about that?
Yeah, so that's spun out of the University of Western Australia and the Harry Perkins Institute of Medical Research. It's so founded around 2004, 2005. It was a private company for about a decade, and then it's floated on the stock market in Australia. And it's just completed 2 phase 2 trials in focal segmental glomerulosclerosis and diabetic kidney disease. So 2 forms of kidney disease. One's an orphan disease, and one's obviously more prominent with diabetes. So that's very exciting. It's very exciting particularly when you spend your life working in HEC293 cells, you then collaborate with very clever people that work on preclinical models to look at the work you've been doing in cell systems, and then you work with very clever clinicians and wonderful patients, and actually see your theories that have come through those model systems actually bear fruit in reality. And we still have patients taking those medications after the trials are finished with special access. So clearly their doctor feels that it's making a difference, and certainly we have clinically significant data to say that we are seeing improvements in kidney function. So that's very exciting, and what's particularly humbling is when you get phone calls from not patients, it's actually the wife or the mother of patients who are desperate to find therapies for their loved one, and they ring you up and they say, well, can we get into this trial? And this is why we do research. It's very humbling.
How did you get involved with the company? I'm more interested into the little anecdotes. Did you always want to be involved in so you mentioned it, so your work is focused on improving human health on the long term.
Mm-hmm.
A lot of academic researchers unfortunately don't get to do this, to be involved in clinical trials, be involved in a more biotech type of setting, so that they can see their work go into the clinic and go into patients. How did you get involved with this company?
Well, I didn't intend to. I was happily beavering away in the academic laboratory with my colleagues, because again, it's a team sport. It's not an individual sport, and we developed an approach that could actually enable us to understand some of these receptor function interactions, and a way of looking at this in a different way. And it was really a case of, well, this could actually be commercialized, and this could actually be taken forward and be used as a discovery engine to find these novel interactions. So I had a choice. I could either work with the company and take this forward, or move off and do something else. And so I thought, well, no, I'm quite keen to see this through. And it was exciting. And it's been very good, both from the point of view of helping us get closer to patient care, but also from funding. We're not constantly having to chase grant funding. We have a diversification of income, and I would encourage any young academic that is looking to start a career to think about different ways of funding their laboratory, and working with companies, with biotech companies, accessing these different sources of funding. It's a good way of ironing out the inconsistency of grant funding, shall we say.
Right. And since we're talking about grant funding, what's the status of grant funding in Australia? Everyone in the US knows that it's getting harder and harder to get funding in the US, in Canada as well. How does that look in Australia?
Yeah, it's becoming harder and harder to get funding for fundamental research, but there is a lot of funding available for translational research. So if you have a clinical focus, if you have a commercial focus, it's becoming easier in terms of there are more funding schemes available. But it's different, because if you want to access clinical funding, you obviously need to work with clinicians. And if you want to access commercial funding, you need to work with industry partners. And it's a different skill set. You have to interact with industry. You have to understand industry's needs. You have to understand the whole commercialization process, not from the point of view of being a CEO, but from the point of view of more of a CSO. So you need to understand enough about the process to go and speak to people, have a management team around you, people that understand business and regulatory affairs and all of these things that we don't really think about as molecular pharmacologists. And so it's a different skill set. And so for fundamental researchers that have been very, very skillful at writing grants and publishing papers, for them to suddenly be told, well, actually, no, you need to go and talk to industry partners. You need to engage with them. You need to pitch to them. You need to get venture capital. These are very different skill sets and a very different world.
Do you feel that your experience with the company that you mentioned before gave you access to developing these skill sets?
Absolutely. And, I've spent many, many years traveling around the world, giving talks to rooms of executives who are interested in different things. And after a while, you get used to that. And in some respects, I prefer it, because if I build a relationship with a partner and they want to work with me and they are willing to commit funds to my project, and as a consequence of that, I can go to leverage a grant from government with a higher success rate, I prefer that than putting in a grant to a panel which has a 10% success rate, and it may just be on that day. There were so many fantastic grants that mine just didn't make the cut. And I think I actually prefer being able to interact with the people that I'm getting the money from.
Of course. How did you get ready for that? How was your first presentation to, key stakeholders who had the checkbook on their desks?
I mean, I've always done a lot of public speaking, even from being at school. So I've never had a problem with public speaking. I think if you're nervous about public speaking, that is something you can learn and practice. And the more you do it, the more it becomes second nature. I think really it's a case of the more you have these conversations, the more you understand. It's about empathy. It's about understanding, what does the person sitting on the other side of the room want from this discussion? And it's about the win-win. And I think at the end of the day, everybody involved in, whether it's the pharmaceutical industry or academic research, we're all interested in treating patients, improving patient lives. Whether people make money from that or not, that's actually secondary. If the patient gets a benefit, then people will make money. So let's make sure the patient gets the benefit, and then everybody gets what they want.
I was asking this mainly for our audience to kind of give an idea to junior scientists and young scientists on how to think about these problems and how to move ahead and not think just about writing grants and sending them to government organizations, because as we mentioned before, it's getting harder and harder to get funding for basic research. And sometimes the starting point in my mind, the thought process is that if I hadn't gone out of the lab and haven't worked in a biotech company, I would have never understood how these processes go. So it's very important to diversify, be open, and try and learn new things. And it may be feeling, or it may be scary in the beginning, but you just have to keep at it and do it.
Yeah,
and
absolutely.
I love that you said that, the whole goal is to think about what does the other person in front of me want, and how can we work together so that we can make it a win-win situation.
Absolutely. And I think it's possible to get internships and placements, and any of these opportunities you can take to spend time with a company, then take them. I think it's not wasted time. It's all good life experience, and these are the skills that we need to take forward. And I think it's also important, if you're doing a PhD, to realize that a PhD is not just a training in working at a bench and writing a thesis. It's so many other things. And in fact, one of the things that I remember back when I was doing my PhD is that we got to go on a week-long residential course to actually understand teamwork skills, and we thought this was another one of these waste of a week, weren't we, in the lab? But it was a fantastic experience, and it taught us that we have so many skills we're learning when we do our PhD, whether it's about time management, people management, if we work in a team, managing a budget, often resourcing. These are all business skills, and these are all skills that they might be given different buzz terms in different sectors, but at the end of the day, you are learning skills, and those can be transferred. And people talk about transferable skills. Well, PhD is 3-4 years, how long it takes you. It's a training in transferable skills, and I think people need to realize that. And I also think it's important that when people finish their PhD, whether or not they go on to do a postdoc, there are so many amazing avenues that they can take their career. They don't need to stay in an academic laboratory. They can go into industry, they can go into teaching, they can go into politics, they can go into business. There are so many avenues that they can take. And the PhD is great training for that, regardless in which direction they go. The analytical skills you learn with a PhD are second to none.
I could not agree more. I think a lot of PhDs and I can count myself in that group, when I was doing my PhD, thought that you can only do basic science in an academic lab and be at the bench. And the goal of the PhD is just to learn how to pipette and how to think scientifically. But now that I've kind of evolved away from that, I realized that you could be doing anything. So somebody with an MBA is maybe good at business, but will never be as good as you are as a PhD at science. But you can learn the business skills as a PhD without having to go and do an MBA. And
Absolutely.
This is something that not a lot of people realize. And I hope that the PhD students and the postdocs listening benefit from our conversation at this point when it comes to these transferable skills. Getting back a little bit to science, so I was looking at your Google Scholar page, and I've seen a lot of papers that say BRET, NanoBRET. Speaking of technology, how did you get into developing these tools to better understand GPCR and beta-arrestin interactions and in the field, and to contribute to the field?
Yeah, it's always been something that we've been keen to do, is to push the envelope. Understand how we can look at protein-protein interactions, complexes, how we can actually use the bioluminescence resonance transfer, which if you've been Michelle's lab, I'm sure you're very familiar with, and really push the envelope. And for example, the ligand binding assay with NanoBRET, which is fantastic, and people are adopting that more and more now. The CRISPR system. So I need to credit Carl White, who's really driven that, a postdoc in my lab. And I think, and obviously Michelle has done a huge amount in this area as well. To develop these systems. And what I love as well is I go around the world, and so many people are using BRET now. A few years ago, there were only a handful of us around the world that were using it. It was very much a niche technology, but now pretty much every GPCR lab, I think, uses BRET, which is pretty incredible, actually. And I think it's really become the go-to technology for molecular pharmacology, and that's very exciting. And just to know that I've played a little part in that is really nice.
It is no secret that I'm a BRET fan and I'm a BRET person. So every time there is any project that involve protein-protein interaction, understanding the function of something or a conformational change in something, the first thought I have is, let's just do some BRET on it.
Absolutely.
And
why not?
I think of course, and I think it gives, what I really enjoy about it is that it's done in live cells. You can, adapt it to 96 well plates, 384 well plates, and you can get an enormous amount of data in a very short period of time. And it gets you to the next level where you can actually go beyond the technology, but get the answers that you're looking for as a scientist.
Absolutely. And I think the real-time aspect is critical. The number of times we see very interesting profiles. And if you were to do an endpoint assay, you would completely miss the point because you've missed the peak, or and a lot of the time you get a signature, and you can compare those signatures and see a lot of nuance. So I think the real-time live cell aspect, as you said, is critical, and why I really like the approach.
Again, I could not agree more. I was having a similar discussion last week with Sam Hoare, who is a consultant at Pharmacology, and we were talking about analyzing this data. So he presented at the Dr. GPCR Summit. He gave a talk about how to analyze kinetic data when you're looking at GPCR activation. And I had shown him two of my publications, one in 2011, one in 2016. And we were looking, and he told me, so you did time courses? And I said, of course, because if you don't look at what happens live during a period of time, how can you dissect all of these traces? Unfortunately, I didn't have the skills to analyze the data. So actually, we're going back right now and analyzing my time course data with his method of analysis to see if we can get any new information out of there.
Very nice, very nice. And I think biosignaling is a good example as well. A lot of biosignaling is probably has a kinetic component, and people have not seen the time window, and you can actually get very different bias depending on the time point you choose. So whenever anybody shows a web of bias or these spider plots,
Yeah.
I always like to ask, at what time point was each of those done at?
Exactly. And it's so interesting because you had mentioned that one of the receptors that you've been working on other than the angiotensin is CCR2.
Mm-hmm.
That's one of my favorites. And I think it gave, so it, as you know, and I'm pretty sure the listeners also know that it binds multiple natural chemokines. And I was a little bit disappointed when I worked on CCR2 because I was trying to see if there is any bias there, but all I was able to show is that there is a rank order between of efficacy between these chemokines. But still, I'm pretty sure that there is something that we could have dug deeper and seen over time, those biases that happen.
Yep. Still lots to do.
Yes, absolutely. So you've worked in academia, you have experience working with a biotech company, getting compounds, getting tools up and running. Do you think biotech companies and pharma is still interested in working on GPCRs?
Yes, I do. I think a lot of pharma companies are looking at data science and looking at mapping signaling pathways and very much moving more towards this sort of looking for new targets. But I do think the industry goes in waves, and I think they've never gone away from GPCRs. I think GPCRs are an important target. As you know, and other people on your podcasts have said, there aren't that, even though there's a very large number of GPCR drugs out there, they actually target a very small number of receptors. And I think there's still an awful lot of potential for targeting GPCRs. I think we've still got a lot to learn in terms of how GPCR is complex and which other proteins are involved and the allosteric modulation that results from that. And I think there's a lot of nuance that we can still learn, which will enable us to target with higher specificity. So I think there's still a lot to do. I think we just need to change the way we screen and we profile, rather than doing more compounds and just getting larger and larger sets. We just need to change the way we're looking at things.
Could you elaborate on how you think we should change the way we're looking at things?
So I think looking at complexes is one example. If whenever I give a talk, I always talk about traditionally screening has happened with receptors in isolation, but in reality, in the body, these receptors are complexing. They're interacting with the cytoskeleton, with other proteins, with other receptors, and that will change their conformation, that will change their ability to bind, whether they're actually interacting at the cell membrane or inside the cell. There's a lot we still have to learn. And I think if we understand those nuances, we can then find new ways to screen and profile with that new knowledge. And I think that then uncovers a whole new potential for drug discovery.
Do you think there are any tools or initiatives that we need to take to speed up measuring these nuances? Are we missing something? Is there more? Well, the answer is pretty much yes. Is there any extra work to be done to get these tools? But what kind of tools you can think of that could help us capture these nuances?
So I think there's two answers to that, really. One is tool compounds that can actually target these complexes in their distinct conformations. If we can work to develop those tools, they may not necessarily be great drugs, but at least if we get some tool compounds to actually understand the pharmacology, that's one aspect. The other aspect is moving towards the cellular systems, the organoid chip type systems. And I'm one of my hats is deputy director of the Australian Research Council Centre for Personalised Therapeutics Technologies. And in that centre, I'm working with some very talented people across the country. So that centre is spanning Melbourne, the University of West Australia, and Monash University, and people developing organoid systems and organoid chip systems that can start to look at systems that are human, but obviously not patients. So moving away from a hectenoid 3 cell more towards a more physiologically relevant system.
I think that's every basic scientist's dream, is not to have to respond to reviewers saying,
yes,
well, is this physiologically relevant? And yes, I think we all agree that hex cells are a great tool, but it's not enough. And having, a more, I want to say more specific, a system that's closer to what happens in the body, without having to, using humans as guinea pigs is very important.
Absolutely.
That's amazing. So I know we've talked about this just a little bit earlier about, young scientists and junior scientists and what they can do to, wear multiple hats like you're wearing. Any other advice for junior scientists who want to contribute to the field? I think it's always welcome from people who wear so many hats as you do.
I think the key is to seek good mentors. I think it's very, very hard to do it on your own. And I think if you can work with people that are happy to give you introductions, if somebody's invited to speak but they can't make it, if they're happy to offer that invitation to you, whether it's reviewing for journals or whatever it might be, having people that look out for you in your career, I think is very important and will become increasingly important. And I think something in the current situation with COVID-19, I spend every day on Zoom, and I'm connecting with people that I know and I've got relationships with, and so we can connect on Zoom, and that's absolutely fine. But I think if I was a young researcher, I'd find it very, very difficult because I can't bump into people at a conference. And I think as senior researchers and senior academics and leaders, we need to be actually looking out for the younger researchers who don't have those introductions, who are missing out on those introductions, and making those connections for them. Because I've spent a long time in Western Australia. We all were locked down for a little while, so everybody was Zooming. So even though I was in London, it was fine because it might have been 4 o'clock in the morning or something, but at least I could be on the same Zoom call as everybody else. But then as soon as people started to go back to having face-to-face meetings, they would still have you Zooming in, but then you were the person and everybody else was in the room, and they just forgot about you, and you couldn't hear the questions, and they forgot to pass the microphone around, and then, let's go for drinks afterwards and carry on talking. It's like, that'll be me hanging out now. So I think we need to realize we're in a new world. We're probably going to be doing Zoom conferences for quite a while, at least for another year, I suspect, in a lot of cases. And in some respects, that's wonderful because my travel budget is certainly healthier than it was last year. But those connections over a coffee, over a beer at the end of a conference, we're not getting those. And I think we just need to be aware of that.
I think, as you mentioned, Zoom has become, I have a 5-year-old who knows how to navigate Zoom, go on a Zoom call.
Absolutely.
Because of COVID. So I think we're getting younger and younger people getting on Zoom and know what Zooming means. It certainly has its advantages. I think a lot of people, since there were so many GPCR related conferences, but I think other conferences that are virtual, it breaks down barriers. It gets rid of, travel budgets, especially if you're a trainee and you need to ask for permission to go to these conferences, but it's not the same thing as sitting, in front of each other, having dinner, or feeling as a student or as a trainee, feeling awkward at a conference where it's dinner time and you're sitting at a table and the keynote speaker just sits down at your table and you have to talk to them. And at some point you realize that they're just as human as you are.
Absolutely.
but they're just more experienced, and you can learn so much from them.
Absolutely.
So before I leave you, I have two last questions. We've talked about science. We've talked about the fact that you wear multiple hats, and having the ability to work in a big team is what allows you to be able to wear multiple hats. Were there any aha moments during your career trajectory as a scientist that showed that, surprised you, or gave you the indication that you're in the right place at the right time?
I think working with dimerics and just as we reached every milestone from that, particularly when the trial results came out and they were positive, and we were actually seeing the protein levels in the urine going down in the patients. And I think, we spend a lot of time publishing papers and coming up with theories and writing about our arrestin interactions with our receptors in a HEC293 cell. But to actually realize that we were actually impacting patients, I think I come back to that again. I think, that is really why I think most people get into medical research. And it's really driven me to not only pursue my research, but also help others. And that's why I'm Director of Biomedical Innovation, because I spend a lot of my time mentoring people, helping set up courses, and working with others that are very good at running courses. And I tend to be the one that gets the money, and other very clever people run the courses. But it's all about facilitating others to be able to go down the journey that I've been on. And I realize that it's not an easy journey. And there isn't a set path. And I think often we come out of university and we've gone through our school, we've gone through our undergraduate degree, and we go on and do an honours project and a PhD, and we do a postdoc, and there's a very clear path. But I think this entrepreneurship path in its very nature is not set. And I think I spoke to one person that went through one of the courses not that long ago, and I said, do you know what you're doing, next year? And she said, I have no idea, and it's so exciting. And that to me, that was the perfect answer, because so many young researchers I speak to, they don't know where their next postdoc is coming from, and they're scared, and they want to, they can't see a career path. But I'm seeing people now coming through these training courses that are just excited about the possibilities. This world is opening up to them. And I think that's a great thing, because it is very exciting. We're very well positioned as medical researchers to help society. And I think that's been demonstrated by COVID-19. So I think we should be optimistic about the role we can play going forward.
I love that answer that person gave you. I don't know when I find it very exciting.
Yeah.
A lot of times, again, coming out of my own experiences, a lot of times I felt, especially as a trainee, that it needed to happen the way I had imagined it. And with COVID, what had happened to me in 2020 is that I felt like I was on a train on a set track, and then COVID hit, and the tracks just disappeared. And that's the best thing that ever happened, because here we are today talking to you, and I'm doing podcasts and talking to everyone, a lot of people around the world. And the goal is to interview everyone who works in the GPCR field, and hopefully we'll reach that 1,000th episode on the podcast. And yes, it's frightening not to know what's next, but at the same time, I think the reward or the excitement of the journey compensate for that moment where you realize, well, wait a minute, I'm not on track anymore, or not on the track that I thought I would be on.
Yeah, absolutely.
So last but not least question, you're in Western Australia, the other end of the world. I'm in Boston. If you have any job openings in your team, where can people find you? And do you usually in your team have people from all around the world, or how does your team look?
So we have had people from all around the world, from Algeria, from Glasgow, certainly from the UK, from New Zealand, from Sri Lanka, so from Malaysia, So absolutely from all over the place. Again, the wonderful thing about science is it's about a shared interest and a shared passion for helping people, and that's shared around the world. I'm on the international advisory group of the British Pharmacology Society, and I work with people from every continent, and it's just wonderful. So in terms of job adverts, I mean, I don't advertise positions. I think if you're a young person, you're looking for a job in medical research, the best thing you can do is network, go to conferences when you can, but if not, use the networks you have through your mentors and connect with people, because most jobs don't get advertised. They are by word of mouth. They're through references. They're through interactions at conferences. And I've hired somebody over after a Zoom call before, but generally I hire people after I've met them, at least at a conference. So yes, it needs that personal touch, and I think that's something to always remember.
That's great. So people, I think and you're not the first guest who tells this. I think it's all about finding what you're interested in and reaching out to the people who are doing the science that you're interested in. And even if you get rejected or say, well, I don't have the space right now, or you cannot come to my lab, I think those connections that you form on the long run, if you keep an open mind, they will at some point go around and lead you to the door that you're supposed to go through.
Absolutely. Yep.
Thank you so much, Kevin, for your time. I think I had a lovely time. I love to learn about the multiple hats that you're wearing. The way I see them is that you have this shelf with all these hats, and depending on where you're going that day, you can put on that hat. But I think the take-home message is, go out there, do the best you can, focus on the science, and also think about the impact that you can have. And it's a marathon and a team effort.
Absolutely. And it's been an absolute pleasure. Thank you very much for having me on the podcast.
Thank you. Bye, Kevin. Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Don't forget to subscribe to the Dr. GPCR newsletter at drgpcr.com/newsletter. You can also leave us a 5-star review on Apple Podcast if you think we deserve it, and share our podcast with your colleagues. Until next time, stay safe.